Okay guys, welcome back to our talk about the urinary system, and today we're going to be talking about the formation of urine. So in our body's production of urine, the main goal or function of producing urine is to maintain the composition of the blood, so what components stay in or get out or eliminated from our blood, and by doing that we maintain a constant body temperature, a constant internal body state, which we refer to as homeostasis. Now, the waste products that we're trying to get rid of in urine formation are basically nitrogenous waste products, which means they're waste products that are created as a result of nitrogen products or breakdown or metabolism. And the three main nitrogenous wastes that we produce are urea, which are caused by breaking down and recycling amino acids, creatinine, which are created every time you move your skeletal muscle, so every time you move your arms, your legs, you're producing creatinine that needs to be removed from the body. And finally, uric acid, and this is produced when we're recycling nitrogenous bases from RNA molecules, for example. So those are the three nitrogenous wastes that we're creating as we're producing urine. And there's three steps in the production of urine, and each one of these three steps kind of takes place along different parts of the nephron, but there's no real cut and dry. So we have first glomerular filtration, then we have tubular reabsorption and tubular secretion. So we'll go through the definition of each one of these now before we start talking about the physiology of the nephron. So filtration occurs in the renal corpuscle, and you guys can remember that the renal corpuscle is made up of the glomerulus and the Bowman's capsule together. Okay, so if we look at the glomerulus, we know it's a series of blood vessels, small arteries, and they have, if you look at the close-up here, these little tiny holes in them that we call fenestrae. The word fenêtre means window in French, and this is where we get the word. So these little fenestrae are tiny pores that can help us filter the blood. So the filtration actually occurs across the glomerular membrane, and then the stuff that's filtered out collects in the Bowman's capsule, and we call that the filtrate. Now, nutrient reabsorption is going to mostly occur in the proximal convoluted tubule, okay? But we've got reabsorption happening in different places along the nephron. Although the PCT is mostly responsible for reabsorption, it can happen all over. Now, when I'm saying reabsorption, what I'm talking about is taking stuff from the filtrate and adding it back into the interstitial fluid that surrounds the nephron, or very often you'll find that called the peritubular fluid. Peri just means around, and tubule means tubule, obviously. So when we reabsorb stuff, we take it out of the filtrate, which is inside the nephron, and we put it into the peritubular fluid that surrounds the nephron. So try and remember that. Secretion is the opposite of absorption. So what we're doing when we're secreting stuff is we're taking it from the peritubular fluid and we're adding it back into the filtrate inside the nephron. So for the most part, the distal convoluted tubule is the main site of secretion. Okay, now that we've been over those terms, let's have a look at how this works, or it looks like the physiology of the nephron. So let's start with the first step, which is filtration. Now, we talked about the blood supply to the kidney, and we know it's got a really healthy blood supply. We've got a branch of the major artery it leads directly from the aorta. So when the blood comes to the kidney, it's under pressure from the heart, okay? So it still has that hydrostatic pressure that's being generated when the heart pumps to send blood around the body. So what we have coming into the corpuscle through the afferent arteriole is our blood, our whole blood. So blood, plasma, everything that's going to be inside of that blood, ions, small dissolved materials, proteins, everything is going to be there. The blood flows into the capillaries of the glomerulus, and it's under pressure, so it actually forces material out of the blood and the plasma. And what happens is all of this extra stuff is what we call the filtrate. It gets collected in the Bowman's capsule, and it leaves to go into the proximal convoluted tubule. What leaves through the efferent arteriole is basically filtered blood. So what we see then are, blood cells, we see big proteins, and that's about it. So everything else gets filtered across those pores. So every little tiny glomerulus does this, and it makes a very, very efficient filtration system. So the pores that we talked about, those little fenestrae, those are going to be between 6 and 9 nanometers in diameter. So that's really, really small. When you think about what we look at under a microscope in the lab, we're talking about micrometers, which is basically 10 to the minus 6. When we're talking about nanometers, we're talking about 10 to the minus 9. So very, very small pores. So the stuff that's going to be collected are going to be very tiny things that we remove and filter out from the blood. Now, once the blood is filtered, the stuff that gets filters out from the blood collects in the Bowman's capsule inside here, and then it leaves the Bowman's capsule to go into the proximal convoluted tubule, and we refer to that fluid then as the filtrate. So here's another look at the glomerulus and the Bowman's capsule. You can see there's pressure being put on the blood, and as a result of that, we get these things moving out of the blood. In this terms, these small solutes, and they're going to move from being collected in the Bowman's capsule and move into the proximal convoluted tubule. Now, these pores, the fenestrae, are quite small. They're small enough to prevent blood cells and plasma proteins. So those things are still going to be held within the blood. They're going to exit the glomerulus through the efferent arteriole, but the rest of it's going to carry on to the proximal convoluted tubule. Now, the glomerular filtration rate is the amount of filtrate that's being produced by your kidneys per minute. So how much stuff is taken out of your blood per minute. And on average, your kidneys, like I said, are very, very efficient at filtration. They produce about 125 milliliters of filtrate per minute. So in a single day, your kidneys will produce about 180 liters of filtrate, and that's a lot of fluid, and that's equivalent to about 70 times the total blood volume. Okay, so 70 times, your blood gets filtered about 70 times per day. Now, all of that stuff that's filtered out, that 180 liters, about 99% of that is reabsorbed. So not only are we efficient at filtering the blood, taking stuff out of it, we're also very good at reabsorbing it so that we don't lose or we don't waste anything that we already have. So incredibly sophisticated physiology, very efficient. Now, once the filtrate moves into the proximal convoluted tubule, this section of the nephron is going to absorb about 60 to 70% of the filtrate. So we usually say that, well, it's a safe bet to say that most of the reabsorption occurs in the proximal convoluted tubule. Now, from this part, we're going to get the reabsorption of organic nutrients, we're going to get the reabsorption of ions, we're going to get some water reabsorbed again, and there's also going to be tiny parts, tiny bits and pieces that are going to be secreted. So it does do most of the reabsorption, but there is in fact some secretion that goes on there as well. So if we have a look at this slide, this is a simplified nephron. So we've got our corpuscle up here, we've got our blood flow. So we get the filtration coming in and happening here. The filtrate is collected in the Bowman's capsule and travels down the nephron. So this is stretched out. So we have the proximal convoluted tubule, the loop of Henle, probably, and then the distal convoluted tubule, and then the collecting duct. So we have all the bits and pieces of the nephron, except it's stretched out. Now, the blood supply here, the efferent arteriole, that leaves with just blood cells and blood proteins. This is what we call the peritubular capillaries, and then the empty space that we'd see here, that's going to be the peritubular fluid. So this capillary, of course, is not going to be straight like this. These capillaries are going to be all wrapped around the nephron, and we'll see a more complicated picture of the nephron a little bit later. And what will happen is the stuff that's reabsorbed will travel from the renal tubule into the peritubular fluid, so the fluid surrounds the renal tubules, and then it'll get recollected into the capillaries and it'll move back into the bloodstream. The stuff that we want secreted will move from the bloodstream and into the tubule and regain or become part of the filtrate again. Once it does that, it's possible for it to be excreted as urine. So this is a simplified picture, but it gives you a good idea about how everything works, how everything comes together. Okay, so we've talked about the proximal convoluted tubule. The next step, we're going to be talking about the loop of Henle. Now, the loop of Henle looks different. We've got two arms, or two limbs. We've got the descending, which is a thin part of the loop of Henle, and then we get the ascending loop or limb, which is, you know, thicker in diameter. And these changes in appearance has a direct effect on its function. Now, surrounded by these two limbs are surrounded by capillaries, and it's also surrounded by the peritubular fluid. Now, the main function of the loop of Henle is to for us to reabsorb water, okay, so that we don't lose any water, because water to land animals is very, very important. So our descending limb, which is thin in shape, is permeable to water. So water can move out of the filtrate in the descending arm. The ascending arm is impermeable to water. So once water makes this turn down here and starts to move back up, the membrane of the cells surrounding this part of the loop of Henle are impermeable, so water cannot escape. It stays in the filtrate. Now, the way that the loop of Henle works is very, very clever, I think. Since water can move out of the descending limb, and you can see that on the picture, we've got water leaving the descending limb. What happens is on the ascending limb, we have active transport mechanisms that push sodium and chloride ions into the peritubular fluid. Now, what that does is it creates a concentration gradient. So we end up with an area on the outside of the loop that's hypertonic, and then the inside is going to be hypotonic. So we get a shift of movement of water just based on the tonicity of the fluid surrounding the loop of Henle. So sodium and chloride ions are actively transported out of the thick ascending limb, and that raises the solute concentration outside the loop of Henle. Now, as a result of that, water moves out of the descending limb by osmosis. So we don't excrete a lot of water in our urine, well, relatively. We save a lot of it because we set up this gradient so that the water is kept in the peritubular fluid, as opposed to being kept in the filtrate where it would be excreted. Now, when we move back, keep moving through the nephron, we get into the distal convoluted tubule, and by the time we get here, we've reabsorbed between 80 and 85% of the filtrate. So there's only 15 to 20% of the initial filtrate left to get to the distal convoluted tubule. Now, once we get here, we'll have some selective reabsorption. So this is kind of where we make the final adjustments to our urine. We take back some of the things that we need, and then we also secrete things in there, and we get rid of stuff that we don't need. So the distal convoluted tubule and the collecting duct, those are the things that put the final touches on the composition and volume of our urine. So the tubular cells here will actively transport sodium and chloride out, so we save sodium and chloride, we don't lose it. We'll also reabsorb calcium ions, so we don't lose those either. And then by hormone control, aldosterone in particular, presence or absence of aldosterone will also help us fine-tune what is lost finally in the urine.